It's late. It's hot. Let's make this a quick one. So as we start to talk about sugar, yes, we should take a moment to remember that sugar became a commodity in the West, thanks to the transatlantic slave trade in no small part. In this story and more, we will cover at some point in the history and social science sections of the content in the future. But yeah, for now, let's get moving. Simple molecules and bonds. We were talking about sugars. We drew ATP, which is the super important molecule that provides all sorts of crazy energy to all of your cells. How to describe it? So it's not so much the phosphate itself that is doing the action. It's more the electrons and the changes in electronegativity that the movement of the phosphate is able to create. So in a way, you can think of the phosphates as like billiard balls. What really matters is the force that they impart, the electrochemical force that they impart when they blast off of ATP, are used by some particular process or are stripped by some particular enzyme or whatever happens to them. But I like to think of it as explosions. Blast off! And then ATP remains until it is refilled with another phosphate, and then the process continues. So that's kind of the way that I like to see it. But the individual components of the molecule are what somehow make this possible on a biochemical and electrochemical level. Yeah. Let's hit it. So, simple molecules and bonds. Number one was ATP. Double check the spelling of adenosine. Oh man, I would have messed that up for sure. Triphosphate. What this is this, but danger. Today, sugar. So there's actually a lot of sugars. It's not just sugar; it's sugars. Yeah. And we're going to start with one of the most common ones, and there's a basic structure that you can keep in mind with sugar, and then there's really only two potential existent structures. One of them looks like this, kind of, and the other one looks like this. So those are your two basic forms and shapes of sugar. Perfection. But I'll give you the actual molecular shape of what I got here. Glucose. Yeah, glucose. Let's look at some glucose quickly. So I'm going to start by drawing the backbone. Nicely done. Then I'm going to draw the pieces off of it. Now you notice we've already drawn something quite similar to this in the ATP. Sorry, always just double checking. This is a slightly longer molecule, sort of dangling off here, which is kind of fun. This oxygen in the middle of the chain is really important. And you can think of this as sort of the most obvious chemical difference in the base layer, or it is the most obvious chemical difference in the base chain, that starts to explain why we have a carbon backbone in this shape. Because we've seen carbon backbones before in many different shapes, but they've all really looked something like this, right? That's been our general sort of shape and structure for carbon backbone. So now all of a sudden, we've managed to loop like this. Now this is possible actually just with straight carbons, but how do you take a branched carbon molecule and bend it into a shape? Well, then you need some sort of polarity to come into play. You need electronegativity. You need electrochemical. Electrochemicals, right? You need electrochemical forces just to come into play in order to bend the molecule into shape in some way, and that can only really happen if you have imbalances in the electronegativities in the molecule. Right? So with carbon chains, they're very nonpolar. They're very stable. Right? So we need to introduce some sort of a much more polar element to our molecules, and here we have all sorts of oxygens providing that very polar role. Right? Oxygens are in H2O. H2O is sort of our default example of a polar molecule. All right, what do I want to tell you about sugars? Let's do this nice and quickly. So, sugars are an energy storage molecule, obviously. Energy storage molecule used by animals and plants to store energy. Animals and plants use sugars in different ways, but they're both using it for energy storage. They take place. They play a role in different chemical reactions. But yeah, what story we already told? Nucleosides. Now, this is some chemical terminology. Are just names, so lots of chemical naming is just names for different subcomponents of the chemical. Nucleosides are then a sugar and a nitrogen-containing base. Nitrogen-containing base. Bases are one of the two sort of basic categories of molecule that you get introduced very quickly into in chemistry. You guys are probably already familiar with acids. Bases are sort of the chemical counterpoint to acids in a lot of actually very sensible ways. So, last but not least, nucleoside. But this is what we're not actually dealing with quite a nucleoside here. We are dealing with a nucleoside, but we're dealing with something a little more specific. We deal with nucleotides. Nucleotides, and most of you are probably familiar with that term already. But that's really just a name for a sugar. Again, but they're attached to a phosphate. Hey! And a nitrogen containing base. A phosphate. So the phosphate makes its reappearance. We love phosphates. Here they are again. Nucleotides are really important because they are the base of DNA and ATP. DNA and ATP. So these are really the two sort of most important basic molecules, if you could even call them basic, that we're going to try and describe. ATP we've already talked about to some extent. DNA we have to really start breaking down. We're also, ATP is our friend. ATP is always there. He's going to show up again and again in all sorts of different reactions of molecules. And if he's not there, then just keep in the back of your mind that he's probably working out, doing something somewhere soon. So I got lots of other little molecules to show you, but I'm not going to do that right now because it's late and it's hot and it's time to go. So, I'll see you guys next time, and thank you so much for watching, and I hope you're enjoying chemistry. Chemistry is pretty cool. I like chemistry. Have a great day.